Okay, is that working?
Perfect.
Okay. So again, thank you for the invitation to speak to you, Ben. What I'm going to talk about today is our recent work on phosphoinositides in the retinas. Before I get started, I'd like to make sure I take care of the acknowledgements so I don't cram them in too fast at the end or forget to do them or not have enough time. Most of the work I'm going to show you today is the work of a longtime research associate in my lab, Feng He. He was helped out by some former postdocs in the lab, Melina Agosto and Ivan Anastasov. Some of the ERG work we did in our lab, some was done in Sam Wu's lab with the help of Dennis Tzu. We had some collaboration with Raju Rajala and Gene Anderson at Oklahoma with respect to type 1 PI3 kinase knockout mice and the cone knockout of VPS34. I list my funding sources here, and I have no commercial interest to disclose. So the topic of today's talk is phosphoinositides, and what I'm showing you here is the structures of the major phosphoinositides that are present in mammalian cells. And the parent compound is phosphatidylinositol itself, for which the head group is this 6-membered polyol that has 6 hydroxyl groups here on the 6-membered ring, and it can be phosphorylated at the 3, 4, or 5 position by the action of the various kinases that I've drawn here. And then the phosphates can be removed by the action of various phosphatases, which are the names shown in the arrows going in the opposite direction. And so these interconversions are constantly going on in our cells, and these enzymes are present in virtually all of our cells. And a number of these, especially some of the phosphatases, such as IMPP5E, synaptogenin, OCRL, have been known to be associated either in human patients or in animal models with retinal disease. A number of them have not been, and primarily because the consequences of their of mutations or deletions of the genes are so catastrophic that they're embryonic lethal. And that's been the case for a number of the genes that we've looked at. And so we end up making cell type specific knockouts. I'm going to talk a lot today about PI3P and the class 3 PI3 kinase that uses ATP to convert phosphotidylinositol to PI3P. Of course, there's another PI3 kinase, the class 1 PI3 kinase that you probably heard a lot more about, which is involved in the formation of PIP3. And both PI3 kinase and PTEN, it's the phosphatase that reverses that process, have received a lot of attention because of the involvement of this class of proteins in cancer. But we've yet to find a really important role for those in the cells that we've looked at in the retina, although perhaps a somewhat minor role in cone cells. So the phosphoinositides are important for regulating membrane trafficking and also for regulating signal transduction. So for example, so what I'm showing here is the structure of the head group for PIP3. So it has 3, 4, and 5 positions, all phosphorylated. But if we have a phosphate only at position 3, as shown here, we get PI3P, which is involved in endosome processing, autophagy, and as I'll show you today, is also important for processing of phagocytosis. PI4P, phosphorylated at this position, is primarily found within the Golgi. It also serves as the major precursor for PI45P2 or PIP2, which has both of these 4 and 5 positions phosphorylated. And PIP2 is very important for signal transduction as the substrate for the formation of IP3, which regulates calcium channels in the endoplasmic reticulum. It's also important in endocytosis, and it plays an important role within the plasma membrane in regulating the activity of ion channels that associate with it directly, such as TRP channels and potassium channels. Now, in order to look at these proteins in the retina, we've had to develop a series of tools. So there have been studies of phosphoinositides going back decades in the retina, but we've been very limited in the kind of information we could extract because of the lack of the necessary molecular tools for looking at specific phosphoinositides. And so we spent quite a bit of time, specifically Feng spent a lot of time developing some of these based on previously published studies from the work of a number of other people in the phosphoinositide field, particularly Tomasz Bala's lab, who pioneered the use of phosphoinositide binding domains. So it turns out our cells are filled with phosphoinositide binding proteins, and they have phosphoinositide binding domains that bind very specifically with high affinities to certain phosphoinositides. So for example, we can localize PI3P by the use of 2 phosphoinositide binding domains from the protein HRS, and we can fuse these recombinant proteins to GST for the purposes of purification, or to GFP for localization within cells or other fluorescent proteins. We can also put an epitope tag on, which we use for ELISA measurements. And we have probes for PI3P, PI4P, and various others. And these proteins are quite specific in their binding. And so here we see this ELISA assay I mentioned earlier, carried out in a spot assay in which we've spotted different phosphoinositides commercially obtained and looked at their binding to these various recombinant binding domains. And you can see that we get very high selectivity for PI3P, PIP2, or PIP3. Now, the tandem binding domains from FAPP1 are slightly less selective for PI4P. They also cross-react with PI3,4P2, but because we have a very good probe for PI3,4P2, which is quite a rare lipid as compared to PI4P, we can always sort out how much of the binding is due to this rare lipid. The assays that we carry out, the ELISA assays for this, are very sensitive. We can detect phosphoinositides down to the range of tens of femtomolar by using the ELISA assays that Feng has developed. These are best done when the PI is embedded in carrier lipids, which they usually are because we just extract all the acidic lipids from the preparations that we make of round outer segments or total retina. And you only need enough of the lipids to cover the bottom of a microtiter well, a single molecule thick monolayer. That's all you need. If you put more than that, the signal saturates. And we can detect as little as 0.001% of total phospholipid. And to give you an idea of what we're trying to measure in terms of the cells, PI3P is present at levels that range from less than 0.002% to somewhat higher. And PI4P is about 10 times or more higher than that, as is PIP2. These are the most abundant two phosphorylated forms of phosphoinositides. And phosphoinositol itself is at relatively low levels in rod outer segments as compared to other cells. It's about 1.5 to 2.5%. Whereas in most cells, it's more in the range of 3 to 10%. So for example, in RPE cells, it's about 6.5%. So the first thing that we did once we had developed these tools was to explore the hypothesis which had been put forward a number of years before, that light induces hydrolysis of PIP2 to form IP3 and diacylglycerol, and to see if we could observe that in terms of a decrease in levels of PIP2 in the light. And while we were doing it, we threw in the other binding domains as well and did all of the assays. And what we found, to our surprise, was that my hypothesis about PIP2 hydrolysis in the light was completely wrong. And in fact, there is a dramatic increase in the levels of PIP2 in the light, and an even more dramatic levels in the increase in the levels of PI3P. Levels of PI4P go up slightly, indicating that there's not a depletion of PI4P when you make PIP2. And PIP3 is present at such low levels that we can barely detect it, and we don't see any change when we go from light to dark. So subsequent work, we decided to focus on PI3P and on PIP2. And I'll begin by talking about our work on PIP2, because that's really in very early stages. We know relatively little about it. This is still a work in progress, and I'll give you a progress report on it, and then I'll tell you in more detail about what we've learned about PI3P, where the work is a lot further along. So the questions that we've managed to answer about PIP2 so far is how much is there in the darkened light, which I've shown you. We'd also like to know where it's located in rod cells and whether phototransduction machinery that we're all used to is required for the light-induced changes that we see, and start to get some idea of what enzymes might be involved. We have not really begun yet to answer the following questions. So what are the mechanisms? If there is a requirement for phototransduction, and we have some suggestion that there is, how do we connect phototransduction activation to the light-induced changes in PIP2? And that gets back to this question of what enzymes are involved. And then very importantly, what are the functions of PIP2 and the light-induced changes in the rods? Why do we need levels of PIP2 to change? Are there any functional consequences of this? So I've already shown you the data for the light-dark changes. The next question I'd like to address is where PIP2 is located. And in order to explore this question, we made use of a technique that was developed a number of years ago in Connie Sepko's lab, which we can express exogenous proteins by injecting DNA into the subretinal space of neonatal mice and basically discharging the capacitor through electrodes placed on either side of the head. And the electric current will drive the DNA into the cells of interest, and we can control which direction the DNA goes by the direction of the current, and which cells it expresses in by selecting the promoter that we use. And when we do this, either the probe itself is fluorescent or we include a fluorescent reporter so we know which cells are transfected. And we can use this to introduce fluorescent markers for specific phosphoinositides, but we can also use this to introduce shRNAs to knock down endogenous RNAs. So we can do cell type specific knockdowns, but I'm not going to show any data of that kind today. So when we first carried this out with our phospholipase C delta PIP2 binding domain, what we found was very bright staining of rod cells, so that works very nicely. But surprisingly, the signal was spread throughout the entire cell as if it were a soluble protein. Now, you don't see the outer segments here. The probe does not diffuse significantly into the outer segments, but it's basically found throughout the inner segments in the outer nuclear layer. And we hypothesized that this lack of specific localization to membranes, where we know PIP2 has to be located, could be due simply to overexpression. So we made use of some work that had been done in Joe Corbo's lab, in which they had fine-tuned the mouse opsin promoter, and they had generated many versions of this, each of which had different levels of expression in mouse retina. And we found one that gave us just enough signal to see, but not enough to flood the cells with soluble protein. And in this case, now we can see in very sparse labeling patches of PIP2 that are found in the membranes of cells. And this staining is now distinct from the signal from a fluorescent marker that we express at the same time, such as GFP, which fills up the cytoplasm. And if we use a membrane marker, such as the cyclic nucleotide gated channel, we can see that there is PIP2 in the outer segment. It's not all over the outer segment. It's located in discrete patches within the plasma membrane, and we know it's in the plasma membrane because of the co-localization, as you can see in this clearly in this bottom panel here with the cyclic nucleotide gated channel. It's also present, and this is just showing more of that. And we also have a marker for the plasma membrane of the inner segment, which is the sodium potassium ATPase here. And if we look closely at that marker, we can see that there's also staining of PIP2 in the plasma membrane of the inner segment. And again, it's in discrete patches. It's not throughout the entire cell. It's in these discrete patches of PIP2 that you can see at higher magnification on the bottom here within the plasma membrane. It's also present in the outer nuclear layer. So the sodium potassium ATPase stains less brightly there, but wherever we see PIP2 staining, we also see the sodium potassium ATPase staining. So it's in the membranes there. If we co-stain with alpha tubulin, we see that it does not seem to be present in the connecting cilia, which are these small structures that you see at the top of this higher magnification view here. And when we see these patches of PIP2, they're on the side of any microtubule structures that we see staining in these cells. So the next question we asked was whether phototransduction is required, which we could look at using the transducin alpha knockout, the GNAT knockout, and mating those with wild type animals and measuring the PIP2 levels in the light and dark. What we find is that in the light, the levels are reduced in the transducin knockout mice. And we see that when we isolate rod outer segments. Now, if you look at the entire retina, you don't see any light-induced changes. So you actually have to isolate the rod cell. Now, when I say we isolate outer segments, we know from extensive work that we've done by electron microscopy and electron cryotomography that what we think of as our rod outer segment prep actually has many connecting cilia attached to the outer segments, many of the cell fragments that we obtain, and there's also large segments of the inner segment. So when we do this outer segment prep, we know that we're getting primarily signal from rod cells, but we can't really say for sure how much of it is in the outer segment and how much of it is in the inner segment. But if we look at the entire retina, we see no effect of the transducin knockout, and for that matter, no effect of light. Okay. Now, finally, what are the enzymes involved? Well, the most likely candidate are one of the various PIP kinase type 1 that are known to convert PI4P to PI45P2. They catalyze phosphorylation at the 5 position. And the most abundant of these, which has been shown previously in the literature, is the gamma isoform, which you can see here stains very strongly, and its levels are indifferent to the presence or absence of transducin, or in presence or absence of light. And we've currently in the process of knocking this out. The global knockout of this enzyme is lethal. So we're in the process of making rod cell specific knockout mice. And it's taken us a couple years to make these, but I think we're going to have them in the next week or so. And this is just to show you that this isoform does stain the entire retina very strongly, but particularly it does stain the inner segments, not the outer segments of the rod cells. And this is as far as we've gotten on PIP2. So there's a lot more to come here. Now, so moving on to PI3P, the interest in this lipid comes primarily from its involvement in 3 different critical membrane recycling pathways. And the first of these is autophagy. Autophagy is so-called self-eating. This is the programmed degradation of internal membranes and cargo to remove damaged components and provide nutrients. And what you have in the case of autophagy is a formation of these interesting sort of U-shaped double membranes that start to accumulate cargo within them in these omega structures, and then they eventually seal to form autophagosomes with a lot of ubiquitinated and non-ubiquitinated cargo inside them, which is eventually fused with the lysosomes and degraded. And when it's degraded, it can release its contents into the cytoplasm for nutrition purposes. Now, the standard mechanism for the generation of autophagy and the role of PI3P in it is shown here. And the way this goes is that there is a multi-subunit complex, and this is for sure, that has as its catalytic subunit a protein kinase, which in yeast is known as VPS34, and I will use the term VPS34 throughout this seminar to refer to this type 3 PI3 kinase that forms PI3P. This is a multi-subunit complex that includes, among other subunits, Beclin-1, and this is regulated in turn by a protein kinase called ULK1, which phosphorylates Beclin and activates it so that it will form PI3P. And under normal, well-fed conditions, this kinase is kept inhibited by the action of mTOR, but various drugs or simply starvation or infection with pathogen can induce inhibition of mTOR, which then leads by inhibition of inhibition to the activation of this complex. Then you get the formation of PI3P, and PI3P then drives the formation of the phagophore through recruitment of a protein called WIPI2 or WIPI2, which in turn recruits something called LC3. LC3 is a ubiquitin-like protein that instead of being attached to protein lysine primary amino groups, as ubiquitin is, gets attached through a similar amide linkage to the primary amino group of phosphatidylethanolamin, and thereby gets tethered into the membranes of these phagophores. And then LC3 2 recruits all of these other proteins here, including a protein called P62, which serves to recruit the ubiquitinated cargo. Now, one reason I'm listing all of this alphabet soup here, which I know is a little hard to keep track of if you don't work on the autophagy field, is that I'm going to show data later with Western blots and immunostaining for markers for antibodies for all of these proteins here. And I'll also show some staining with LAMP1 and LAMP2, which are markers for the lysosome, because the terminal step in this process is the fusion of the autophagosome with the lysosome, which leads to degradation. And this is the one step that, I'm sorry. And we can accumulate lysosomes. We can block this final step. We can block this degradation step by adding chloroquine, which disrupts the low pH in the lysosomes and prevents this fusion process. And so that will be used experimentally in some later slides. But the final thing I want to say in this slide is that when we knock out PI3P through deletion of VPS34, the one step in this process that is unquestionably blocked is this one of the fusion of the autophagosome with the lysosome. And you get accumulation of all of these markers, and you get accumulation of LAMP1 and LAMP2. You do not get prevention of this first step as would be suggested by the standard model here. And I'll also show in our studies with RPE cells that this role of the requisite role for WIPI2 in this process through being recruited by PI3P is also somewhat questionable. Now, another process in which PI3P plays a critical role is endocytosis. So PI3P is found in early endosomes where it's bound by EEA1, which is a classical marker of early endosomes. This happens to be a PI3P binding protein. It has some of those PI3 binding domains that I mentioned earlier. Sometimes those can be used as a probe as well. It's also present in late endosomes, and for which the marker is RAB7. And again, I'll show some data later with staining for RAB5 and RAB7 that shows the co-localization of RAB5 with PI3P and the accumulation of RAB7 in the PI3 knockout, because the common thing that you have between endocytosis and the processing of late endosomes and autophagy is the fusion with lysosomes. So late endosomes that contain cargo that are to be degraded will fuse with lysosomes, and that step is blocked when PI3P is eliminated. And then finally, a process that is not directly important or that we haven't studied directly in rod cells, but that's incredibly important for an adjacent cell layer, the retinal pigmented epithelium, is phagocytosis. PI3P also plays an important role there. And again, PI3P actually is required for the recruitment of LC3 to the autophagosomes. And this phagocytosis is a process that happens on a daily basis. It's subject to diurnal rhythms in which the tips of rod outer segments are shed. So you get a stack of discs that are engulfed by RPE cells, and then they form these phagosomes, and those are, again, are processed by fusion with lysosomes and degradation. And a common thing that all of these three membrane processes have in common, in addition to a role for PI3P, is an endpoint that involves degradation via fusion with the lysosomes. And in all three cases, that particular step is blocked. But in the case of phagocytosis, some of these early steps are blocked as well when PI3P is not present. Okay. So the first thing we asked about the light activation of PI3P was the time course. And what we found is that rather than being some very rapid response to the activation of phototransduction, that it's a very slow process that occurs under continual exposure to light during light adaptation. And this does require light. It's not a function of the time of day, because we've done this at different times of day. And there's always an upregulation in the light and a downregulation of PI3P after prolonged times in the dark. But it does require phototransduction, or at least most of it does, because we don't see it in the transducing knockout. Where is PI3P? So PI3P in rod cells is located in these rather large puncta that you find at different sizes in different parts of the cell. So there are very large puncta that are observed near the outer nuclear layer, and at least some of these are likely to be Golgi, as we've seen in other cell types. We haven't explored that in great detail here, but most of them seem to be distinct from the Golgi, although the Golgi lights up very brightly for PI4P. And then as you move from the outer nuclear layer towards the outer segment, within the inner segment, there are decreasing sizes of these puncta that we see. And though we haven't figured out what all of those structures really are. Now, there are two PI3 kinases that have received a lot of attention in the literature in the last several years. And these are the ones I mentioned before. The type 1 PI3 kinase that makes PIP3, as you see on the right here, is also theoretically capable of making PI3P, which is why we had to knock it out to see if it had any effect on the levels of this. But the major enzyme responsible for making PI3P is the type 3 PI3 kinase, which I'll refer to as VPS34. Now, VPS34 is not, when we isolate right outer segments, the VPS34 is not contained within the outer segments themselves. If you see here the Western blot showing different fractions from a density gradient in a rod outer segment preps, you can see that there's very little overlap between the fractions containing rhodopsin and the fractions containing VPS34. VPS34 is a soluble protein, even though it's a lipid kinase, and it is largely found in the soluble phase when we break open the cells and isolate the rod outer segments. The particular allele that we used was one that was generated in Fan Wang's lab at Duke, and she generated an allele that that has a floxed section within the gene that, when excised with Cre, just removes the catalytic core of VPS34. So you still make structural protein. I'll show you that later in a Western blot. But its ability to phosphorylate phosphatidylinositol is eliminated by the elimination of the catalytic core, as you see here in this structure of VPS34 in a simulation of it being docked to a membrane. And we made this with the ICRE75 mice, which we received from our colleague Jason Chen at Baylor, which gives you good expression in rod cells. And of course, as Chris's lab found out a number of years ago, there's some odd things about the ICRE75. They also happen to overexpress R9AP, one of my favorite proteins, but that sort of odd feature doesn't really have much effect on the results I'm going to tell you about today. So you can just think of this as a rod cell specific knockout. And when we analyzed the PI3P levels in the rod cell specific knockout, we found that that in the rod outer segments, there was basically no PI3P left. The very little bit that we could detect above background can be easily attributed to the presence of a small amount of contaminants from other cell types, as compared to the large amount that we see in the flox allele without expression of Cre. And we did similar experiments with the P85 knockout of the type 1 PI3 kinase, and there were no effects on the PI3P levels, and there was no effect on the health of the rods. And that work has been published. In the case of the PI3P, in the case of VPS34 knockout, there is a degeneration. Now the cells, the outer segments form. So in the early stages of development, you form what looks like a fairly normal retina. There's a few aberrations in membrane structure, but rhodopsin is properly transported to the outer segment, so you don't need PI3P to transport rhodopsin to the outer segments. You don't need PI3P to form outer segments, as has been previously suggested. You can form it just fine, but very quickly the cells begin to degenerate. So just by one and a half months, you're already seeing a dramatic shortening of the outer segments, a loss of nuclei from the outer nuclear layer, and by 3 months or so, virtually all of the rods are gone. Okay, so this is a pretty aggressive retinal degeneration. And in these cells, you also see an accumulation by electron microscopy. You can see the accumulation of aberrant membranes that have these sort of empty looking spaces here that indicate lipid droplets. And you can see that there are many sort of multiple membrane structures. There's membranes within membranes in these membrane aggregates that we find in the inner segments of the VPS34 knockout rods. And you can see a close-up of that on the bottom here. You can see that there are membranes within membranes, which is characteristic of autophagosomes, although autophagosomes are generally somewhat smaller than this. And there's an accumulation of autophagy markers, including LC31, LC32, which is the form that's covalently modified with phosphatidylethanolamin, P62, ATG9, and so on. We can see this by Western blot. We can also see this by immunostaining, where we see the accumulation of LC3 puncta that are completely missing in the wild type mice, but we see lots of these puncta. And we can look at LC3 either by using an LC3 antibody, as shown in the top here, or as shown in the bottom, panel D here, through the use of an LC3 GFP fusion that's expressed in a transgenic line that we've mated with these. And you see that although there's tons of GFP LC3 present in these cells, that it doesn't form these nice puncta that we see in the knockout mice. And the autophagy markers, LC3, ATG9, and P62, all co-localize as expected for formation of correct autophagosomes. So PI3P, based on these findings and the electron microscopy, PI3P is not required for the formation of autophagosomes, but it's necessary for the processing of autophagosomes. So when you delete it, what you end up with is an accumulation of autophagosomes. And you also see, as you see here in this with the LAMP1 and LAMP2 staining, you also see an accumulation of lysosomes. Now, the VPS34 complex itself, including its subunit partner Beclin-1, are still intact. There's maybe, if anything, a slight decrease in Beclin-1, although the VPS34 levels are okay. But this VPS34 form is lacking the catalytic domain, and that's what's giving rise to all these phenotypes. So it's a very nice model in that it's a fairly clean deletion of a catalytic activity. You also get an accumulation of ubiquitinated proteins. So there's a wide range of ubiquitinated proteins of various molecular weights. And even though you get a decrease in the levels of free ubiquitin, and these punctae co-localize with the autophagy-related membrane. Now, there are some ubiquitin puncta that do not co-localize, but all the ones shown in arrows here are GFP, LC3 positive GFP puncta that also contain ubiquitinated cargo. And so these autophagosomes have all of the markers of true autophagosomes. They contain the molecular markers, and they look like autophagosomes by electron microscopy. Here you see them merged here. You also get accumulation of lysosomes, as indicated by the LAMP1 puncta here, but these do not co-localize with LC3. Now, this is found in the knockout mice. In contrast, in the wild type mice, we can also force accumulation of LAMP1 by adding lupeptin, which prevents degradation of cargo within. It is a protease inhibitor that will inhibit the proteases within the lysosomes, and we can add rapamycin to stimulate the accumulation of autophagosomes, and the combination of these gives you accumulation of LC3 positive punctae. And these LC3 positive punctae also are positive for LAMP1. So in the presence of PI3P, you still form these LC3 puncta, but now they fuse with the lysosome. So this result shows unequivocally that the step that is blocked by PI3P removal is the fusion of the autophagosomes with the lysosomes. You also get accumulation of the late endosomal marker RAB7, presumably for the same reason, failure to fuse with the lysosomes. And the RAB7 late endosomes are distinct from the autophagy-related membrane. For the most part, you do not see any co-localization of LC3 with RAB7. So these are distinct processes, although they both involve PI3P. So that's pretty much it for the knockout in rod cells, and this was all published in Scientific Report a few years ago. We then moved on to bipolar cells, and I'll just tell you briefly that, again, we find that the deletion of PI3P leads to cell type specific degeneration of the cells. We knock these out on bipolar cells using a PCP2 promoter-driven CRE, which is expressed in rod bipolar cells and a subset of cone bipolar cells. It's also expressed in Purkinje cells of the cerebellum. So these mice have a severe ataxia because of degeneration of the Purkinje cells. But you lose the on bipolar cells, particularly the rod on bipolar cells, which are stained by PKC alpha up here. The cells that do not express PCP2 are not lost. So loss of the PCP2 expressing cells does not lead to death of the other cells. And you see not a complete abolition of the ERG, but you see a diminution in the amplitude of the scotopic and photopic B waves due to the loss of bipolar cells. And this is a rather slowly, in contrast to the rod cell knockout, this is a rather slowly progressing disease. And in these cells, again, we see accumulation of autophagosome markers. We see LC3 and P62 accumulate in the knockout cells. And these are rod bipolar cells as marked by PKC alpha. And there's fewer of them, but those that exist have these P62, large P62 accumulations within them. And you can also see the autophagosomes accumulating by electron microscopy. So then finally, for the last part of the talk, let me move on to the retinal pigmented epithelium, where we can, in addition to autophagy and endocytosis, we can focus more on phagocytosis, which is an important function of the RPE. And so when you see these large hexagonal looking sort of RPE cells, these are images of flat mounts of the RPE. And you can see that in wild type mice, if we express our PI3P marker, that GFP without the PI3P binding domains is just expressed as a soluble protein throughout the cell. But there are puncta that are formed when we have the PI3P binding domains, as viewed either in a section as shown here or in flat mount as shown here. You see lots of accumulated membranes. And as it turns out, these are a mixture of autophagosomes, endosomes, and phagosomes, because PI3P is involved in all of those processes. And when we look at the knockout mice, they don't have any PI3P. We see the green fluorescence, but we don't see any punctiform. And you know that these are knockout mice because we have the CRE antibody. Now, the way that we made the these knockout was to use a bestrophin promoter-driven CRE, and bestrophin promoter has some advantages and disadvantages. It's specifically expressed in RPE cells, but it's not expressed in every RPE cell at all times. And so we get mosaic expression and mosaic knockout. That has the disadvantage that we have to infer from the phenotype or from CRE staining whether a particular cell has the knockout or not. But it also has the advantage that we can compare wild type cells to knockout cells right next to each other in some cases, or at least in the same retina. Now, one thing we also noticed about PI3P, so we looked at the PI3P localization, what these puncta are, and we largely did this in cultured RPE cells. So this is one advantage of working with RPE cells, is that there are a number of cell lines which are not quite the same as RPE cells in vivo, so we have to keep going back and forth between the two, but they're quite easy to manipulate. You can generate a lot of experiments quite quickly. If we look at markers of the endoplasmic reticulum, those do not co-localize with PI3P or a marker GM130 of the cis and trans Golgi. But if we look at markers for the, cis and medial Golgi, but if we look at the trans Golgi marker, TN46 or RAB5, there is co-localization, and the endosomal markers RAB5 and RAB7 also co-localize with PI3P. Now, if you induce the formation of autophagosomes in these cultured cells by adding chloroquine, which I mentioned before, increases the pH in the lysosomes and forces accumulation of autophagosomes, then you see accumulation of these PI3P positive puncta, and you see accumulation of LC3 positive puncta, but they don't truly co-localize. Within the PI3P aggregations, you see these black holes that are devoid of PI3P, and those are precisely the locations where you find the LC3. Now, in the literature, you'll find this sort of pattern described as co-localization, and a lot of the models that you'll see in various review articles describing how LC3 is recruited by PI3P through the action of WIPI2 to these membranes is based on describing this sort of pattern as co-localization. But in reality, if two proteins can be resolved from one another by confocal microscopy, given the resolution of confocal microscopy, they cannot be in the same molecular complex. They cannot be physically bound to one another, although there is clearly some correlation between the two. So I think there's a lot yet to be learned about how you go from PI3P accumulation to LC3 recruitment, and the standard model is perhaps oversimplified, to say the least. Now, when we made knockout mice using the bestrophin promoter, it does cause cell death in patches of the RPE, those patches that are positive for crease staining, as shown in the upper left here. And you see by electron microscopy that there's a lot of distortions of the membranes, not only of the RPE, but also the underlying outer segment membranes. And this is a relatively slow process compared to what we see with the rod cells. And again, as I mentioned before, it's mosaic. So here you see by RPE65 staining. Here's a wild type mouse. You see nice uniform staining. You see these nice hexagonal patterns that you get from ZO1, which is a marker for the junctions between the RPE cells. And you see these large patches of dead cells or missing cells that have died and been carried away in the knockout RPE. And adjacent to them, you find cells that presumably are relatively healthy that have RPE65 and ZO1 junctions. So you get these accumulated membranes that you can see by electron microscopy that, as I'll show you from staining by various markers, consists of these abnormal membranes are frustrated late endosomes, autophagosomes, and phagosomes. All three of these membranes accumulate. So here are the frustrated late endosomes, which stain for RAB7. And again, this is a nice case where you can see a couple of knockout cells immediately adjacent to some cells that presumably still have PI3P because they don't show the phenotype. Now, as far as autophagy, we see the same accumulation of classical autophagy markers that we see in the rod cell knockouts, P62 and LC3. These co-localize and these co-localize. So we've also looked at phagocytosis, which is an extremely important process for RPE cells. And one tool that we've made use of for looking at phagocytosis is a line we developed a number of years ago, which has a knockout of a GFP fusion with rhodopsin into the mouse rhodopsin locus. And the heterozygotes of this mice are morphologically quite normal. They give relatively normal photo responses, but it's very easy to visualize their shed disc tips, as you can see here. And you have to do this at the right time of day because of the diurnal rhythm I mentioned, because in wild type RPE, these are processed, and even though you can see them right after they're shed, a few hours later in the day, they're gone because they've fused with lysosomes and been degraded. Now, when you look at the knockout mice, you see a very different story. You see, no matter what time of day you look, you see this dramatic accumulation of autophagosomes. All of the staining is due to this rhodopsin GFP that is accumulated at very high levels in phagosomes in all of these knockout cells. And you can look at these by electron microscopy. You see many aberrant membrane structures, but some of them are filled with what you can see here are not individual stacks of discs, but multiple stacks of discs, which is one reason that these are so large in the knockouts as compared to the phagosomes that you see in the wild type, because these seem to fuse with one another and form these very large structures with many stacks of discs within them. And this is just a section viewed from the side where you can see the same thing, that you see very few phagosomes in the wild type, but when you have knockouts here, you can see an accumulation of these puncta, of these large phagosomes accumulating around the knockout nuclei. Now, interesting, the LC3 is recruited to phagosomes as it is to autophagosomes. And remember, in the case of autophagosomes, LC3 recruitment still occurred in the absence of PI3P. That is not the case for phagosomes. So here we see on the top some phagosomes in VPS34 wild type mice. These still have PI3P, and these co-localize with this red staining for LC3 here. See these where you see the arrows. Okay, but in the knockout mice, you see the accumulation of these rhodopsin puncta. You see the accumulation of these LC3 puncta, but they're completely separate. They are not co-localized. So in the case of phagocytosis, you absolutely need PI3P for the recruitment of LC3 to the autophagosomes. ATG9, which is a critical player in autophagy, does not seem to play a role at all in phagocytosis. It is not recruited either in wild type or knockout mice to the phagosomes. So even though phagocytosis has been referred to as LC3 mediated autophagy and such terms, it's really a completely different process from autophagy. And there's only a few players that they have in common, such as PI3P, LC3, and of course fusion with the lysosomes. Now in these these VPS34 knockout RPE cells, in addition to the accumulated phagosomes, we also see accumulated autophagosomes, which are distinct from the phagosomes by their staining with LC3, P62, and ATG9. So they have all the classical hallmarks of autophagy in the absence of PI3P. And so you can easily distinguish them from the phagosomes. And they also contain ubiquitinated cargo. And again, as in the knockout rods, they do not fuse with the lysosomes. Now, let me just treat briefly on the question of WIPI2, WIPI2, which has been proposed to be the mechanism which recruits LC3. WIPI2 binds directly to PI3P, but when we look at the phagosomes, which we know, I've shown you on the previous slide, these are decorated with PI3P and with LC3 too. These do not bind WIPI2. There are puncta containing WIPI2 within the RPE, but they do not co-localize with the phagosomes. And that's also true, of course, in the knockout mice that do not have PI3P. So that is a little bit funny. If WIPI2 can bind directly to PI3P and recruit LC3, why is that not working in phagocytosis where both PI3P and LC3 are present? So we looked at that in RPE cells, and what we found is that if you add chloroquine to spur the formation of autophagosomes that are labeled with both WIPI2 and with PI3P, we can add an inhibitor of VPS34, a chemical inhibitor. And when you do that, you see a loss of PI3P signal. You can see that these puncta get dimmer and dimmer as you go on in time in this top panel here. But WIPI2 stays there the whole time. It's totally indifferent to the loss of PI3P. WIPI2 is bound to the same membranes in these autophagosomes as PI3P, but not through the medium of PI3P. Something else keeps it there. And then finally, the LC3 recruitment to the autophagosomes does not require WIPI2. So we can knock down WIPI2 using shRNA in cells that we co-label with DS red. So all the red cells here are missing WIPI2 as indicated by the absence of staining with the green label of WIPI2 antibodies, as you see in the central panel here. And you can see here through the merge that these red cells are all lacking WIPI2, but they're not lacking puncta for LC3, that when we accumulate autophagosomes through addition of chloroquine and blockage of lysosome fusion that we see in the WIPI2 knockout cells, we still see an accumulation of LC3. So WIPI2 recruitment to PI3P is not required, is not necessary or sufficient for the recruitment of LC3. And so that's basically where we are with the PI3P in the RPE. So I'd just like to conclude then by summarizing what I've told you today, which is that light increases PI3P, PI4P, and PIP2 dramatically in rods. PIP2 is located primarily in patches in the plasma membrane. I didn't show you the data on PI4P, but it's in the Golgi, and there's some in the plasma membrane. PI3P localizes to distinct membrane compartments, and that includes endosomes, trans-Golgi, phagosomes, and autophagosomes. A knockout of VPS34 and PI3P causes cell death in rods, RPE, and on bipolar cells, and these are due to this death is due to defects in processing of late endosomes, autophagosomes, and phagosomes. And there's also an accumulation of lysosomes. And what these have in common is the blockage of fusion in the knockout of all of these different membranes with the lysosomes. And some surprises, some results we have that are sort of perhaps still a bit controversial and disagree with certain things that are in the literature, is that PI3P is not required for the formation of outer segments or rhodopsin transport. Recruitment of WIPI2 and LC3 to autophagosomes does not require PI3P. I didn't show you the RAB11 story, but that's not required either. And recruitment of LC3 does not require WIPI2. The type 1 PI3 kinase and PIP3 are not important for rod cell survival. And phagosome processing in outer segments is not a subset of autophagy. It is completely distinct pathway, although there are some points in common. It does not involve ATG9, P60 or P62. And in the case of phagocytosis, LC3 recruitment requires PI3P, but it does not in the case of autophagy, and the engulfment of shed ROS tips does not require PI3P. So I think I'm pretty much done there, and I'll be happy to take any questions.
If you have any question, please drop me in chat that you have a question. You don't need to write the question, just to say that you have something to ask. Ted, thank you so much for a wonderful presentation and again so much work on this important molecules. I wanted to also advertise next week we will have Henrik Scholl from Basel, who is going to tell us how to cure blindness, not only in mice, but in humans. So hopefully you will join us. So the first question goes to Dr. Taylor.
Hello?
Hello.
Yeah, successful.
Did you ask the question already? I was muted here.
No, you just ask a question what you wanted to ask.
Okay, so Ted, that's a fabulous talk. Thank you so much. And I'm wondering, since there's oftentimes an interplay between the ubiquitin system and the autophagic pathways, so when you basically shut down fusion that ends up in lysosomal pathways, are you chasing a lot of substrates into the ubiquitin pathway? And do you see differential activities of the ubiquitin proteolytic pathway?
So we have not looked at proteasome activity. We see an accumulation of ubiquitinated proteins and of ubiquitinated puncta. So clearly, any activation of the proteasomal pathway is not sufficient to overcome the blockage of autophagy. But whether it's stimulated or not, we actually haven't looked into it. That's a good suggestion to do that.
Okay, let's move to 3.
Excellent talk, Dr. Wensel. I have a simple question. It was my understanding that PI3P is required for phagosome genesis and genesis of the omegasome. So how do you, so I was expecting that in your GFP-LC3 model that you would not see any signal at all because there is a failure of phagosome genesis, but you still do clearly observe immature phagosomes. So how do you explain this observation? Thanks for the talk again.
Okay. Well, what I can say is that, I think the standard model is a little bit oversimplified. Certainly, PI3P is recruited into the vicinity of the omegasomes and the autophagosomes, but as far as we can tell, it's not required for the formation of the autophagy-related membranes because they have all the markers, even though PI3P is not there. So I can't tell you exactly what takes its place when it's not there, or how exactly WIPI2 and LC3 are recruited, but they do get recruited without PI3P. And I'll just tell you that a number of people in the autophagy field don't really like me saying that, but that's what we observe. And if you look at other people this knockout mouse has been, this cell type specific knockout of VPS34 has been done in some other cell types, and they basically see the same thing. So we're not the only ones.
I have another follow-up question to that. So what do you observe in electron microscopy? Are those phagosomes open? Do they fail to close completely? Or are they microtubule bound yet? Because it's really important for those microtubules to undergo anterograde motion on microtubules to fuse with lysosomes.
So I'm sorry, could you repeat the question?
So what is the structure of those phagosomes? Have they fully closed yet? And if they have, is it a failure of those phagosomes to be recruited to microtubules, which is what is required to fuse with lysosomes?
So that's a good question. We see overwhelmingly closed membranes. So we see membranes within membranes, but we don't see an accumulation of unclosed omega structures. But that could just be because they're rare. We don't see any microtubules associated, so apparently that is blocked, and perhaps that's the critical step. We don't really know that, but they certainly are not associated with microtubules, and they also don't transport properly, so they stay more or less in the same region of the cell. So normally there's a dramatic translocation of the phagosomes, and you can't really see that in a flat mount, but that's, if you look in sections, you can see they kind of stay where they form, very close to where they form. So that may indeed be a problem of association with the microtubules.
Yes, it was striking that most of those immature phagosomes were in the myoid region.
All right, let's move to Dr. Swarb.
Yeah, hi Ted. Fascinating talk. I learned a lot. I think this problem is sort of, I think in my mind at least, quite complex. I think your phenotype is so interesting, and PI3 kinase does so many different things. I was wondering how you are going to sort out how the cell death finally happens and what pathway is critical. Shri asked part of the question actually, which I was going to ask, what is the structure of these autophagosomes? Whether the problem, I mean, this is involved in endocytosis, this is involved in autophagy, there's all sort of recruitment and recruitment of in cargo. I mean, I'm not sure, whether you have this kind of accumulation impacts the cargo accumulation as well somewhere. And I was just wondering that in vivo, it's somehow a little bit hard to resolve these questions. And I was wondering if you have some thoughts about using maybe cells or iPSCs or some fibroblasts or something derived from these mice to look at that question, some of the mechanistic question. For example, even autophagy flux. I mean, you may need to see whether it's the initiation or the degradation or what is going on. So maybe some sort of cell culture model derived from these mice could be useful. I don't know what your thoughts are. I'm just very curious to know.
So that's a good point. So there are many more questions unanswered than we've managed to answer. I would agree with you on that. And certainly cultured cells are sort of more easy to figure out than the situation in vivo, although one could argue sort of in some ways less interesting or relevant. But we certainly have an interest. I mean, one thing we're planning to explore is primary culture of RPE cells from the knockout mice. Although, it's actually easy to knock out VPS34 using, using CRISPR in cultured cells. For example, we could do that in RPE cells, and we've done a lot of work with cultured RPE cells. We can also do primary cultures of RPE from the mice. And so I would agree that there are a lot of experiments we can do that way, and we're moving in that direction.
Thanks.
And I have a question related to this VPS34 knockout. You mentioned that you have a patchy death of RPE cells. That's a great model to look what happened to photoreceptors in this area. Did you look into that, this relationship between RPE and photoreceptors? Yes.
As you might expect, if you have a patch of multiple dead RPE cells, the rods underneath them, first of all, have dramatically shortened outer segments, and a lot of them are dead. But it's patchy enough that you don't get a statistically significant decrease in the thickness of the outer nuclear layer. So cells die, but they just die here and there. So, yeah.
Yeah, but there was also an idea that RPE cells, once it's dead, the area that occupies is taken over by neighboring RPE. Did you look on the sizes of RPE in those mice as well, those that survive?
Yeah, so I don't know if you noticed, but in some of the images I showed where you have the ZO1 staining, you do deviate from this nice, more or less uniform cell size with these nice hexagonal structures to these very expanded sizes. But of course, there's so many nuclei, some of them are stained for Cre and some are not. You sort of lose track of which cell is which, but there's definitely an expansion of cell size that takes place in the vicinity of the Cre positive cells.
I think this will be, again, very important for therapies if you look into preservation of RPE and the relationship to photoreceptors. Dr. Taylor, you had another question, I guess.
Yeah. Hi, I think maybe Anand already scooped me on my question.
That's what he does.
But my line was partially out, so I'm not sure I heard it, the full answer. And that had to do with, are you identifying the cytotoxic species?
No, I would say if you have a good suggestion for how to do that, I would like to know. And that does impinge on Anand's question. I mean, how you go from the observations that we have to cell death is a big question, as in my opinion it is in most cases of photoreceptor or RPE cell death. We can show you all kinds of things that are wrong, but which one is actually critical for causing the cells to die. I mean, especially in these animal models where cell death takes place at some time during a several month period, it's a little tricky to do that, because if you knock something out, I mean, if you can knock something out and rescue it, that's a pretty clean experiment. But I don't know how we would apply that in this case. But if you have any suggestions, I'd be delighted to hear them.
Well, maybe you could do something like a reciprocal experiment, like where you build up ubiquitin conjugates, say just interrogating them, and then reverse that, release that buildup and see what the fate of the cell is when you don't have, say, VPS 34 or comparable autophagy, regulators. Well,
yeah, that's
you have that. In a way, you have that data. I think even in your VPS 34 knockout blot, if I remember seeing, you had diminished ubiquitin, but diminished free ubiquitin, but enhanced buildup of conjugates. So you almost have that. It seems like you almost have that system in your hand.
Yeah, I agree. If we could incorporate into that some manipulation of the proteasome system, I think we could learn a lot. That's a great idea. Thank you.
All right, let's move to Chris Sander, Dr. Sander.
Hi, Professor. Really fascinating talk. I got a little confused by your last your summary slide there. Did you guys see a reduction in the light of PIP2 and generation of IP3, or?
No, we see an increase in PIP2 in the light.
Okay, so right.
Okay.
So does that sort of answer the?
No, we have not looked at IP3, and there is some phospholipase C present. So whether you are generating IP3 or not, we haven't looked at that. Some other groups have, but I think there's a lot more that needs to be done there.
Right. Yeah, I was just wondering if you had sort of answered the controversy that Wensel, or that Professor Anderson had gone into, that kind of thing, with whether or not decreases that they saw in PIP2 would be generating IP3, that kind of thing. So,
yeah, I mean, I think it's possible that you can. I mean, we've seen in cultured cells that you can activate pathways that stimulate phospholipase C. You can generate IP3, but the steady state levels of PIP2 do not change substantially, and that appears to be because there's some coupling of the synthesis of PIP2 to the hydrolysis of PIP2, so that even though you generate a lot of IP3, PIP2 levels stay fairly constant. There are some situations where we've seen that before, and that could be what's happening in rods as well.
and all these enzymes are co-localized, it seems, like in the inner segment, sort of, as well.
Well, they are all present in the inner segment, but to be, co-localization, to my way of thinking, they have to be actually right next to each other.
Right.
In contact, and we're not really sure about all of that. I mean, there's a lot of translocation that goes on.
Gotcha. Thank you.
All right, we had a wonderful conversation here. We have some suggestion from Brother Anand in chat, Ted, for you. The final question will go to Alex. Alex is from Poznań, and that's a good news. Poznań is in Poland. And then final comments will go to Professor Imanishi.
Yeah, hi. I wanted to ask about the PIP2 staining that you showed in the rods. So could you comment on why the staining was so scarce and not many cells were actually stained there? And also I wanted to ask what time of day actually this staining represents, and whether you maybe looked at some changes like in the time course of the day for this type of staining?
Yeah, so the answer to your second question, unfortunately, is I don't know the answer to whether it depends on the time of day. We have looked at it on, with and without light exposure, but I mean, the problem, your first question sort of defeats the second one, because it is so sparse, it's sort of hard to get great statistics on this. Now, there are two reasons for the very sparse labeling that I showed. One is that the way we do the electroporation, there is generally sparse expression. So some cell, but usually in one field we can see several cells that have taken up the DNA. The other reason, though, is that since we see the staining only in small regions of the plasma membrane, and it's not always the same region, even though two cells that are next to each other may both have the DNA, they may not be stained in the same place, and so we may not capture them in the same field in the microscope. And so it's sort of the probability is low because of the sparse staining. You multiply that times the probability of finding them in a given field, even if they do have the DNA, and you end up with a very sparse looking staining, only a few cells here and there. And so this is sort of a challenge of the work. We have to collect lots and lots of images to get any decent statistics on it and to compare animals that had, I mean, and the electroporations are also somewhat difficult to make reproducible, in terms of numbers of cells that get transfected.
Yoshi, all yours.
Yeah, thank you. Yeah, so yeah, Ted, thank you very much for sharing a great story. Can I ask one more question then?
If we have time, I've gone kind of long here, but
very brief question. So, your another favorite protein, did you take a look at the transducing alpha subunit, which is considered to be the substrate or that's degraded by autophagy in rod photoreceptor cells? So Dr. Ferguson and Zach's group,
yeah, so we do not see any accumulation of any of the phototransduction proteins. We actually see, but the problem is we see loss of them because the cells are dying, right? So if you do a Western blot, all the phototransduction proteins are going away because the cells are dying. But whether there's buildup in the cells that survive, I don't know.
Thank you very much.
Thank you, Ted.
Okay, thank you.
We'll see you each other on Friday next week.